to study pollen germination on a slide. Materials required: nutrient medium. For making the nutrient medium, we take 100 ml of water and dissolve 20 mg of potassium nitrate, 30 mg of magnesium sulfate, 10 mg of boric acid, 10 g of sucrose, 10 mg of calcium nitrate. We dissolve all these substances together in water and our nutrient medium is ready. Because we are carrying out this experiment in the laboratory, so we require the nutrient medium so that the pollen germination can take place. Now we would require a fresh flower, cover slip, cavity slide, needle and the brush. You can see there is a concavity in the cavity slide. There is a depression wherein we are going to put our nutrient medium. With the help of a dropper, we put 2 to 3 drops of the nutrient medium in the depression. With the help of the brush, we dust the pollen grains over the nutrient medium. Gently we are going to dust the pollen grains. Then we place the cover slip over the nutrient medium, and there is a particular way to place the cover slip, making an angle of 45 degrees and gently placing it so that there are no chances of air bubbles. This way we hold the cover slip at an angle of 45 degrees, and slowly with the help of the needle we place the cover slip over the nutrient medium on the slide. Now this slide we will leave it for some time, for at least 15 to 20 minutes, so that the germination process can take place. I've already prepared one slide. This I'll keep it under the microscope. First, I'll adjust the microscope and keep my slide on the stage of the microscope, fix it with the help of the clips and then observe it. I'm able to see that the pollen grains have started to germinate. Not all of them, but few of the pollen grains are germinating. Aim of the practical is to prepare a temporary mount to observe pollen germination. Principle: pollen germination is the male gametophyte. It contains the male gametes. Pollen grains germinate and form pollen tubes after they get deposited by the process of pollination on compatible stigma. Pollen tube, made up of cellulose, is an extension of the inner wall of pollen grain intine. It emerges through one of the germ pores and passes through tissues of stigma and style to reach the ovary. The generative cell also passes into it. It soon divides into 2 male gametes. Pollen grains can also be induced to germinate in a synthetic medium. Because we are carrying out this experiment in the laboratory, the pollen germination experiment, therefore we require the nutrient medium. Materials required: 10 g sucrose, 10 mg boric acid, 30 mg magnesium sulfate and 20 mg of potassium nitrate, 20 mg of calcium nitrate, 100 ml of distilled water, cavity slide, cover slip, brush, needle, microscope, dropper, beaker and a fresh flower which has the mature pollen grains. For example, China rose, balsam, jasmine, lily or Trasidentia. Procedure: prepare the nutrient medium for pollen germination by dissolving 10 g of sucrose, 10 mg of boric acid, 30 mg of magnesium sulfate and 20 mg of potassium nitrate, 20 mg of calcium nitrate in 100 ml of distilled water. Using a dropper, take some nutrient solution and put 2 drops on a clean glass slide, cavity slide. With the help of a brush, sprinkle mature pollen grains on the drop of nutrient medium. With the help of the needle, place the cover slip on the drop. After 15 minutes, observe the slide under the microscope. When we observe the slide under the microscope, then we are able to see that several pollen grains germinate and the pollen tubes come out through the germ pores. The pollen grain is uninucleate. It has one nucleus in the beginning. At the time of liberation, it becomes two-celled with a small generative cell and a vegetative cell. In the nutrient medium, the pollen grain germinates, the tube cell enlarges and comes out of the pollen grain through one of the germ pores to form a pollen tube. The tube nucleus descends to the tip of the pollen tube. The generative cell also passes into it. It soon divides into two male gametes. We'll draw the diagram of the pollen. We'll label XII, the outer layer, intine the inner layer. Will show the germ pore and also will show the pollen tube coming out through the germ pore. We'll also label the generative cell and the generative nucleus and the vegetative nucleus. In the longitudinal section of the flower, we can also show precautions to be taken. Collect the pollen grains from a fresh flower only which has mature pollen grains. Place only 1 to 2 drops of the nutrient medium on the slide, not more than that. Now let's see some Viva questions. What is pollination? Transfer of pollen grains. What are the different factors responsible for pollination? Air, wind, water. And some animals are also responsible for pollination to take place. Which part of the flower contains pollen? Anther. What happens to the generative nucleus in the pollen tube of the germinating pollen grain? It divides into two to form two male gametes. What does the pollen tube carry? It carries the male gametes. Can you explain as to why some pollen grains fail to germinate? Because might be they are not viable. Why do we use sucrose as the medium of pollen germination? So that the pollen gets the food, gets energy from it. Can pollen grains of one plant species germinate on the stigma of other species? No, it cannot germinate. Hello students, in this particular video we are going to talk about experiment 2 and 3, which are to study plant population density and plant population frequency by quadrat method. So one by one we are going to first see the density and then the frequency. So let's start with first experiment, that is to calculate the density. So what is the aim of our experiment? To study plant population density of different species in a given area by quadrat method. Materials required: string, scale, nails, hammer, notebook and pencil. So we require these things when we are doing the experiment in the field. Otherwise, if we are doing the experiment in the laboratory or during examinations, then ma'am is going to give you a sheet in which different plant species are drawn by different symbols, and you have to calculate the number by counting those species. Theory: population. First of all, let's see what is population, what is density, how do we calculate population density, what is a formula, and what is a quadrat. Okay, so first of all, what is a population? It is the group of individuals of a particular species occupying a particular area at a specific time. The number of individuals in a population is not constant. It gets affected by birth rate, death rate, immigration and emigration. This you have studied in your theory also. So therefore the population keeps on increasing or decreasing in a particular area. So how do we actually find the density? So density of a population, it refers to the size in relation to some unit of the space on observed time. So density, it represents a number of individuals per unit area. Quadrat is the name given to a sampling unit and area of a definite size. For example, when we do the experiment in the field, then we generally take a quadrat which is 1 meter into 1 meter in size, 1 meter square in size. So how do we make that square? We take nails, we hammer the nails in the field and then we tie a string and we calculate the total species, we count the number of the species which are falling inside that square. So this unit of area, the sampling unit, it is called as a quadrat. Now, how do we calculate the density? Total number of the individuals of the species divided by the total number of the quadrats studied. So, population density of different plant species, it can be calculated by laying quadrat of suitable size and recording number of individuals of each species which are occurring in that quadrat. So, what is the procedure? First of all, we are going to select a suitable area for study and hammer the nails on the site in the form of a square. Tie the string on each nail so as to obtain a square of 1 meter into 1 meter in size. Make 6 such squares in the area. It is very difficult to identify the different plant species taxonomically. So what do? We identify them by naming them as A, B, C, D and F. Note down the number of individuals or species A, B, C, D, E and F in each square to calculate the density of the species in given area. Okay, so this is the procedure when we are doing the experiment in the field. But during your examination, your ma'am is going to give you this kind of a sheet in which the different plant species are represented by this symbol. Let's say this is one particular, this is species A. Okay, this is species B. Here are the names of different species written. Here is species C. This is species D. Okay, this is species E and this is species F. So here different species are represented by different symbols. So what is actually to be done during your examinations? You have to draw a square. Okay, any suitable size you can take, or you can ask your ma'am what size of the square you have to take. Okay, so you have to draw with the help of a pencil at least 6 such squares, and you are going to count the different plant species which are falling under that square. Okay, these are 6 squares, and you are going to count the different species. For example, these two species, species D, these two species are of species D. This one, they are species, and this one, these are species F. So this way you are going to count and write the observation. This is how the observation table is to be made. First of all, you're going to write name of the species. Here we are naming them as species A, B, C, D, E and F. Now we are going to write the different quadrats, name of different quadrats, quadrat 1, 2, 3, 4, 5 and 6, and we have to count the species A, if which is falling in quadrat 1, it comes out to be 12. For example, species 2 in the second quadrant 14, third quadrant 16, then 11, then 9, then 8. Okay, then we have to add them up and the total comes out to be 70. We are going to now write the total number of the quadrats studied. It is 6, and density, how it is to be calculated? Density is equal to the total number of the individual of the species divided by the total number of the quadrats studied. And for species A, it comes out to be 11.6. Now, for the next quadrant, species B, next species, the number of individuals in each quadrant, it comes out to be 29 divided by 6, 4.8. Next species C, 17. Total number of the species is 17 in all quadrants, and species C is not present in quadrant number 4, so we are going to put a cross sign. Okay, then total number of the quadrats studied 6 and it comes out to be 2.8. This way you are going to calculate the density. Now by looking at this density, we can find out that this is the highest density. Species A is highest in number and species E is lowest in number. So we can say density of species A is highest and density of species E is lowest. So how do we calculate the result? What is the result? What is the conclusion? The population density of species A is highest while the population density of species E is lowest. So in terms of density, the field is dominated by species A. Precautions: select a thickly populated area. Only those plant species should be counted whose roots are inside the quadrat. Carefully count the number of the individuals of each species. So this way we have calculated the density. Now we are going to talk about the population frequency. Again, it is very simple. The aim it is to find out to study plant population frequency of different species in the given area by quadrat method. Material required, it remains the same. Theory: the population is a group of individuals of a particular species occupying a particular area at specific time. The number of the individuals in a population is not constant. It gets affected by birth rate, death rate, immigration and emigration. Now, what is frequency? The frequency of plant population is the number of times a plant species appear in the given number of sampling areas or the quadrats within a study area. Now, what does it mean? It means that, let's say we have studied let's say 4 quadrats, and in the 4 quadrats the species is present in first, it's not present in the second quadrant, it is present in the third quadrant, it is not present in the fourth quadrant. So we have to just check the its occurrence in different quadrants. For example, out of 4 quadrants it is occurring in 2 quadrants only. So the frequency could be calculated up as 2 upon 4, and it is to be multiplied by 100 because we find out the frequency percentage. So 2 upon 4 into 100, that means 1 upon 2 into 100 is equal to 50%, is equal to 50% is the frequency percentage of that particular species. Okay, so percentage frequency is equal to total number of the quadrats in which the species occur into 100, divided by total number of the quadrats studied. So it's usually expressed as a percentage. So population frequency of different plant species can be calculated by laying quadrat of suitable size and recording the occurrence of individuals of each species in that quadrat. This is the procedure. The procedure is same as we have studied earlier about population density. Here we have to check the occurrence. We don't have to count the number of the species. We just have to check whether it is occurring in a particular quadrat or not. So note down the total number of the quadrats in which the species A, B, C, D, E and F are found to calculate their frequency. So here we are going, what are we going to do? In your practical examination, you are going to get this kind of a sheet, and ma'am is going to give you only one experiment, either second or the third one. Either you are going to calculate the density or the frequency. You are not going to get both of them. So this way you are going to make 6 different squares, and you have to just check whether a particular species is present in that square or not. Okay. So for species A was present in square 1, quadrat 1, 2, 3, 4, 5, 6. So total occurrence of the species in the quadrats is 6. That means it was present in all 6 quadrats. And how many quadrats we studied? 6. And then what is the frequency? Frequency is equal to 6 upon 6 into 100 is equal to 100%. Okay, so occurrence of species in quadrat was 6. Total number of the quadrats studied was 6. So frequency is equal to P by Q into 100. Species B, for species B again we are going to calculate. So species B is, let's say it's present in all 6 quadrats. So the frequency comes out to be 100%. And let's say this species E, it is present in only 2 quadrats out of 6. So 2 upon 6 into 100 is equal to 33%. So the frequency of species A and B is highest, which is 100%, and frequency of species E is lowest, which is 33%. So what is the conclusion? Population frequency of species A and B is same, 100%, which is highest, while population frequency of species E is lowest in the given area. Now what are the precautions? The same as earlier. Select a thickly populated area. Only those plant species should be counted whose roots are inside the quadrat, and carefully count the number of the individuals in each species. So this way you are going to do these two practicals when they are going to come in your board examinations. You're going to get only one of it, either the frequency or the density. Of the experiment to make temporary mount of onion root tip to study mitosis. For this, materials required are onion root tip. This root tip of onion, it was fixed in acetic acid and methanol, and then it was kept in one normal HCl solution. Now, onion root tip, we require glass slide, needle, brush, cover slip, spirit lamp, and the stain acetocarmine, dropper, and water. Forceps. First step, we'll take the clean, dust-free glass slide. We'll keep it aside and we'll give several washings to the onion root tip so as to remove the preservative from it. We'll give it at least 3 washes to remove the preservative from it. We'll transfer the onion root tip onto the slide, and we have to make sure that the piece of the tip that we are taking, it is the pointed side from where it was growing. So we have, with the help of a blade, we have cut a small piece of the onion root tip. Now with the help of a dropper, we'll place 2 to 3 drops of acetocarmine stain on the root tip. Just 2 drops of the solution are enough. Now with the help of a needle, we'll put the cover slip over it gently. There is a particular way of keeping the cover slip on the slide. We hold the slide from one side, place it over the material, and with the help of a needle making an angle of 45 degrees, we slowly, gently place it. Now we are going to tap the cover slip very gently with the blunt end of a pencil, or we can use the blunt end of the needle, so as to separate the different cells. We'll light the spirit lamp and we'll give the slight intermittent heating so that it gets fixed properly. After heating the slide, we will squash it again with the blunt end of the needle. We'll tap the cover slip very gently, and we have to make sure that there are no air bubbles under the cover slip. And we have to tap it till it becomes almost transparent. Gently we are going to tap it. Now our slide is ready to be observed under the microscope. We'll adjust the microscope. First we will see the slide under the low power and then under high power. Aim: prepare a temporary mount of onion root tip to study mitosis. Materials required: onion bulb, conical flask, petri dish, scissor, forceps, needle, methyl alcohol, acetic acid, HCl, acetocarmine, distilled water, spirit lamp, microscope, slides, cover slip, and blotting paper. Theory. The cells undergo mitosis, which is the equational division, and it is subdivided into two following phases: karyokinesis and cytokinesis. Karyokinesis, it includes prophase, metaphase, anaphase, and telophase. Cytokinesis is the division of the cytoplasm to form two daughter cells. Prophase. Appearance of chromosome. Metaphase. Spindle formation and arrangement of chromosomes on the equator. Anaphase. Movement of daughter chromosome towards the poles. Telophase. Formation of daughter nuclei. The cells of the onion root tip provides the most suitable material for studying the process of mitosis. You can see the different stages of mitosis: early prophase, late prophase, metaphase, anaphase, and the telophase. In the metaphase stage, you can see the chromosomes, they have aligned themselves along the equator. Procedure. Take an onion bulb and trim off the old roots from its base with the help of a sharp blade. Place the onion in the conical flask containing water so that the base touches the water. Keep it for a week so that the roots start to grow. Cut 5 ml of the tips of the root and put them in a vial containing mixture of 1:3 acetic acid and methanol. This process is called West fixation. Remove 2 or 3 root tips and hydrolyze them by warming to 60 degrees and 1 normal HCl for 15 minutes. Remove the root tip and wash them thoroughly in water. Place a drop of acetocarmine on a slide. Put 1 root tip on the drop and place the cover slip on it. Gently squash the root tip by tapping the cover slip with the blunt end of the pencil or needle until the cells separate and spread out into a very thin layer. Gently warm the slide on the flame for a few seconds. Observe under the microscope and examine the different stages of mitosis. Observation. Under low power of the microscope, rectangular cells with pink nucleus are seen scattered. Under high power of the microscope, the various stages of the mitosis such as interphase, prophase, metaphase, anaphase, and telophase are clearly visible. Precautions. The base of the onion bulb should be in contact with water while growing the roots. Roots should be fixed in morning time. The slide should be warmed gently. DNA isolation. The aim of the experiment is to isolate DNA from the plant source. Here we are taking onion for this experiment. Materials required: test tube with test tube stand, sodium chloride, tilt ethanol, beaker and a glass rod, funnel, pestle and mortar for grinding the food material that we have chosen for our experiment, spatula, glass cylinder, onion. For this experiment I have taken onion. Any other plant material can also be taken. For example, spinach or strawberry or banana can also be taken. And a filter paper. Liquid detergent. So let's proceed with the experiment. The very first step is blending. We are going to blend the onion to a paste. The purpose of blending process is that it degrades the cell walls and the membranes further, permitting the release of DNA. We don't have to blend it for too long as it will break up the DNA fibers. After blending it, then we are going to prepare our extraction buffer. For preparing the extraction buffer, we take a glass beaker. In the glass beaker, we would add liquid detergent to it. 5 ml of liquid detergent I have taken approximately. To the liquid detergent, we'll add sodium chloride. Why liquid detergent is used? Because the detergent and the heat treatment, it degrades the membrane phospholipids and proteins, releasing the DNA. Sodium chloride approximately 2 to 3 grams I have taken. I have mixed it with the liquid detergent. Now I'll add warm water to it. Because the heat treatment by adding water, it would degrade the membrane phospholipids and it would help in separation of the membrane and release of DNA. Slowly, gently by the side, I have added water. The entire process is to be carried out gently so as to avoid the entry of air bubbles into it. We'll mix it thoroughly. This is our extraction buffer. Now it is ready. The next step is the onion paste which we have prepared. We are going to transfer it into the extraction buffer. And after transferring it, we'll leave it for some time undisturbed for at least 15 to 20 minutes so that the DNA can be released and it can come out from the cell. The onion paste is transferred into the extraction buffer and we are going to mix it thoroughly now. The salt is added because the positively charged sodium ions from the salt shield the negatively charged phosphate groups of the DNA molecules, helping them to precipitate out of the solution. Gently we are going to add by the side the leftover amount of water so that the solution becomes 100 ml. We've used warm water for this particular experiment. This extraction buffer along with the onion paste, we are going to leave it for some time, at least for 20 minutes. Now we are going to the next step is filtering out the solid from it. We'll take the glass cylinder and we'll place the funnel into it. We'll fold the filter paper and place the filter paper in the funnel. And gently we are going to pour the solution into the funnel. And we have to make sure that there is no air bubble. The solution that we get in the measuring cylinder, we will transfer it into the test tube, into a clean test tube, and then we'll add chilled ethanol to it. And we have to ensure that it is added drop by the side so as to avoid any air bubble and it forms a layer at the top. Why we used chilled ethanol? Because it slows down the breakdown of DNA which would occur if a high temperature is maintained. And ethanol, it will precipitate into the upper layer. The DNA, which is the white material, it would be clearly seen in the alcohol layer. We'll leave it for some time. For 10 to 20 minutes, we'll leave it. I have already made a solution in which I've already added the alcohol, the chilled ethanol I have already added and which I've already kept for 15 to 20 minutes. As you can see clearly on the top in the ethanol layer, you can see the entangled mass of DNA. The white substance that you can see floating at the top, this is DNA. The thread-like structure, entangled mass, we are able to clearly see. DNA isolation. Aim: to isolate DNA from available plant materials such as spinach, green pea seeds, papaya, onion, etc. Materials required: plant materials such as spinach, green pea seeds, papaya, onion, etc. 5 gram liquid detergent, 2 gram sodium chloride, chilled ethanol, 95% isopropyl alcohol, distilled water, test tube, funnel, filter paper, test tube stand, pestle and mortar, and the beaker. Principle: deoxyribonucleic acid DNA is a genetic material in plants and animals. The recombinant DNA technology has allowed the introduction of DNA in other organisms including bacteria, yeast, animals and plants. These organisms are called as genetically modified organisms. Thus, recombinant DNA technology involves isolation of DNA from a variety of sources. Procedure. Make the paste of onion with the help of a pestle and mortar so as to break the cell's walls. In a beaker, mix the liquid detergent, salt and 100 ml warm distilled water. Stir gently, trying to avoid making too many bubbles in the mixture. This is the extraction buffer. Add the extraction buffer to the onion paste and mix again. Leave the onion extraction mixture to incubate for 15 minutes. After 15 minutes, filter the extraction mixture through a filter paper. This will remove all the solid material that we don't want. We would be left with a clear liquid. Transfer this liquid in a test tube. Pour 10 ml of chilled ethanol carefully down the side of the test tube to form a layer on the top of the content. Let it stand for some time. Observation. At the interface of alcohol and onion, we would see white cloud-like substance forming. The addition of ethanol to the solution causes DNA to precipitate. The DNA fibers appear as white precipitate of very fine threads. Precautions. The plant material should be clean so that there is no dust particles sticking to it. All the glassware should be thoroughly dried and cleaned. The chilled ethanol should be added slowly by the side so as to avoid the entry of air bubbles to it. This is the double-stranded DNA that you can see in the image. We have to draw the diagram also when we are doing the practical. The backbone of the DNA is formed by sugar and phosphate, and purines and pyrimidine bases are present. Based on this, you can get the viva questions based on the structure of DNA. You can be asked the precautions to be taken in the viva.